So today's topic I'm really excited about because it's relevant for every human who wishes to live a long and healthy life. And I'm fairly confident that's everyone here today. And as clinicians, and certainly for everyone here at Atria, what drives us is the pursuit of the ideal that everyone's health span should equal their lifespan. So from the perspective of our public health mission, aging is also a critical risk factor for a variety of human diseases, from cancer to hypertension, to diabetes, to neurodegenerative diseases like Alzheimer's disease. So the ultimate goal then is to identify and validate molecular targets for intervention that extend human health span and human lifespan. However, given the fact that we already live a fairly long time, the length of the human lifespan makes it difficult to assess those interventions using death, of course, as an outcome. So we need a quick readout, if you will, if we're going to test the effectiveness of an intervention on aging. In other words, we need a biomarker that accurately correlates with the diseases and the structural and functional manifestations of aging so that we can measure the effectiveness of any intervention. So that's what makes this presentation and our guest today so important. Dr. Steve Horvath is a professor of human genetics at the David Geffen School of Medicine at UCLA and a professor of biostatistics at the UCLA Fielding School of Public Health. Dr. Horvath obtained his PhD in mathematics at UNC and a PhD in biostatistics at Harvard. He's an aging researcher and bioinformatics expert whose research lies at the intersection of epidemiology, chronic diseases, epigenetics, genetics, and systems biology. He proposed an epigenetic clock theory of aging and published actually the first large-scale study on the effect of lifestyle factors on epigenetic aging rates. There are lots of salient features of the epigenetic clock, which I'm sure he's going to go into. Dr. Horvath, thank you so much for taking the time today to be with us. We're honored to have you, and we have all been eagerly awaiting your presentation today. The floor is yours, sir.
It's a great honor for me to present to you today at the Atria Institute, which pursues very noble goals. I'm all about open science, making tools available. I will talk about so-called epigenetic clocks. The name clock indicates that we are measuring time, but we are measuring age as opposed to other famous clocks you've heard of, circadian rhythm. Circadian rhythm, in essence, measures hours of the day, morning versus evening, whereas these aging clocks allow us to track the ages of all nucleated cells. We can measure the age of any organ, any tissue. And the hope is, of course, to find deviations from the expected age. For example, if one of our organs is substantially older than expected, maybe that is bad news. My personal interest is to measure aging in people who follow the perfect lifestyle. In other words, it's not my mission to convince people that they should stop smoking. If they don't know it, it's very unfortunate. But I'm interested in helping people who do everything right. They avoid stress, they sleep a lot, they avoid substance abuse, they eat vegetables all day. Everything perfect, but they still age. And I call this aging process that is largely independent of stress factors, innate aging, inborn aging processes. And I'm very much interested in understanding these and, of course, to develop interventions against these innate aging processes. I want to start with an important distinction. Clinicians obviously deal with human organisms, and there are wonderful clinical biomarkers that are routinely being checked. For example, blood pressure, lipid levels, glucose levels. And these are very valuable biomarkers, and they will never be replaced by anything I present today. We will always need them. However, these clinical biomarkers do not apply to cells in a dish. Imagine you want to study aging in fibroblasts or organoid models. So for the latter, for studying really aging at the molecular levels or cellular level, we need these molecular biomarkers. And one of the pitfalls of clinical biomarkers is that it's hard to distinguish whether they measure pathology or truly aging. For example, obesity is clearly a major risk factor for various diseases. However, does obesity really increase biologic aging? And we could have a long discussion about that topic, but it comes back to what I said. Well, if somebody is lean and avoids obesity, well, maybe they're still aging.
Did it work for you?
Innate version. Yeah. Pardon me? Yeah, I also want to mention that strong mortality risk predictors may not relate to root causes of aging. You may have a viral infection or radiation exposure or substance abuse or poisons. All of these stress factors can lead to early death, but maybe they do not accelerate innate aging. And why? Because a person who avoids viral infections and avoids substance abuse still ages. So to measure this elusive biologic age, that's what we all want to have, a measure of biologic age. To measure that is quite difficult, and there's a substantial literature on that topic. Many clinicians will measure biologic age by age-related conditions on the right-hand side, various frailty measures, sarcopenia, lung disease, cognitive assessments, and so on, clinical biomarkers. And there's quite some literature on using these age-related morbidities in various frailty measures or multimorbidity measures. And they're clearly very valuable and clearly very predictive of mortality risk. Pathologists will look at various markers of tissue pathology. And people in my field will look at various omics data. Today I will talk about so-called methylation data, but you can imagine gene expression data, proteomics data, metabolite data, glycome data, many, many different omics data. And I would never claim that any of these data are more important than others. To really assess biologic age on an organismal level, you really need all of the above. You need to integrate them. I, of course, will talk about one of these data modalities today, methylation, but I just don't walk away thinking that I don't think any of the others data are important. As a matter of fact, I measure them routinely in my own research. Now, what I do want to mention is that we are in a very fortunate position in the aging field in that we have a gold standard variable, and that's just age, whereas other fields have to struggle with definitions. We are fortunate that we can say that any biomarker of aging better correlate very strongly with chronologic age. And I will talk a lot about epigenetic clocks, but I want to give you my definition of what I consider an aging clock. Above all, it's really a number. So there's a procedure, an algorithm or a software, and it arrives at a number. And this number could measure chronologic age, your calendar age, or it could measure mortality or morbidity risk. And I make that distinction because when people hear about biologic clock, it has many connotations, but I'm very quantitative about what I mean by an aging clock. So I mentioned, for me, a clock is not some causal molecular process. Mitochondrial dysfunction clearly plays a role in aging, but it's not a clock because I don't have a quantitative measure, or if you have a set of causal genes, various progeria models, Hutchinson-Gilford progeria, there's a mutation. So for me, that's again not a clock. So yeah, for me, a clock measures age and is numeric. I also want to share my axioms of what I call an indicator of biologic age. And I want to mention it because the field has struggled a lot with how to measure biologic age. And so I just want to give you 4 axioms that I feel have to be satisfied. First, I talked about it already. It has an indicator of biologic age must be a quantitative measure that is highly correlated with age. Second, such a measure should apply to all mammalian species. And that axiom reflects that all mammalian species that I'm aware of age. The bowhead whale is the longest lived mammal. The bowhead whale can live for more than 211 years, but still it ages. The third is whatever measure you have has to relate to mortality risk. For example, the abundance of gray hair does not relate to mortality risk after you subtract out chronological age. So yeah, not everything that correlates with age is a biomarker of aging. But conversely, again, I make a distinction. A mortality risk is not the same as an indicator of innate aging. And then the fourth axiom is perhaps debatable, but for me, an innate aging measure should measure a fundamental property of most cells. Why? Because cells age, so it must measure aging in cells. And one of these fundamental properties is, of course, the DNA molecule. And that's what made telomere length such an attractive measure. It measures telomere endings, the endings of chromosomes. And so it was a very attractive biomarker for that reason. The problem of telomere length is that it does not quite satisfy axiom number 3. It actually only estimates mortality risk weekly. You really need huge sample sizes to detect an association with mortality risk. Now, moving away from these axioms, I'm moving to now the topic of methylation, cytosine methylation. And I've worked on cytosine methylation for the last 12 years. You may ask, why cytosine methylation? Why not gene expression? Why not proteomics? And the reason is cytosine methylation has 3 extraordinary properties. First of all, you can develop aging measures that apply to the entire life course, from prenatal samples, meaning early development, all the way to centenarians. And that's quite unique because many biomarkers of aging really apply to aging, right? Middle-aged people, but you could not apply them to children. Different for cytosine methylation. The second remarkable property of methylation is that you can build these pan-tissue clocks or multi-tissue clocks that measure aging in all tissues. And that's also remarkable because I'm not aware of another omics data type that allows you to do it. For example, proteomics clocks have been developed and they seem to work quite well. but they only apply to blood. There's no evidence that the same proteomics clock applies to liver tissue or skin tissue. Yeah, and then the third aspect is even more remarkable than the above two, which is methylation allows you to build so-called pan-mammalian clocks. So these are clocks that apply to all mammalian species. And this is so remarkable because, as many of you know, we have cured most diseases in a mouse model, but these findings often fail to translate to humans. So there are often substantial differences between a rodent model and a primate model. And just crossing the species barrier has been very difficult for most biomarkers. But fortunately, it is relatively easy to use cytosine methylation to build clocks that apply to multiple species. So with that, I show you a typical Excel sheet that I look at in the morning. So the different columns are different people. These are blood samples. And then the rows are cytosines. And people with a good eye will notice that all the numbers lie between 0 and 1. And so what it means is that 0 means none of the chromosomes are methylated at one location. So let's say I have one location on chromosome 1, and I can form the proportion or estimate the proportion of all chromosomes that are methylated. For example, if you have 100,000 chromosomes, then the upper left number would mean that 93% of these chromosomes are methylated at this location. And when it comes to the number of rows of this Excel sheet, the sky is the limit. Human beings have 28 million cytosines. I would say at least a quarter of them correlate with age. Age has a profound effect on cytosine methylation, which is why I like it so much. It's a wonderful approach for building biomarkers. And statistically speaking, epigenetic clocks are prediction methods, linear regression. What you learned in a biostatistics class, a regression model will get you there. It's a multivariate regression model. The dependent variable, the y, is either age or measure of mortality risk. And then all the covariates are these cytosines. And to mention a few buzzwords, machine learning, artificial intelligence, penalized regression models. But these are just buzzwords because it is as simple as your introductory biostatistics class, just a regression model. And the approach incurs what I call the original sins of the construction. So epigenetic clocks are based on a mathematical optimization procedure, least squares regression, right? So you try to minimize the error in the prediction. And that incurs a cost because we don't have a ready biological interpretation, right? In a multivariate model, you may find 350 cytosines have been selected, and you then need to make sense of what do they measure. So that's the first original sin. And contrast that to telomere length. Telomere length has a beautiful interpretation. We know telomerase, we know a lot of the biology. And however, telomere length is very inaccurate, so there are these trade-offs. Do you want something very accurate but hard to interpret, or the other way around? The other original sin is then multiplicity of clocks. You can build literally millions and billions of clocks by slightly changing the cytosines or the training set. And so in certain ways, we need to solve that by creating benchmark data. Now, I want to mention that we have many different types of epigenetic clocks, and I distinguish them by generation. So the first generation of epigenetic clocks were defined as predictors of your age, your chronological age. My UCLA team developed the very first clock, which was a saliva-based clock. You spit in a cup and we measure your age. And it was a curious observation, but we weren't sure whether it was biologically exciting. And then 2 years later, I published the so-called pan-tissue clock, the clock that applies to all organs at the same time. And that publication generated quite a lot of interest because what I mentioned, the difficulty of developing biomarkers that apply to multiple tissues. But the first generation clocks had a limitation, which is they weren't particularly good at predicting mortality risk. They do, no question. If you have 3,000 people, these clocks will predict mortality risk. But maybe not in a way that is clinically interesting. And to address that, we developed second generation clocks based on blood. So these are blood-based clocks, and they are quite accurate for estimating mortality risk. Accurate enough that life insurance companies, for example, have started to become interested in using these clocks for stratifying people by mortality risk. And these second generation clocks are probably most interesting to you as clinicians. So some of these, like Grim Age, which was named after the Grim Reaper for a reason. It's our best mortality risk predictor. But this Grim Age clock was already used in a clinical trial. So people use these clocks already in clinical trials of anti-aging interventions. I will also talk about so-called third generation clocks. These are these multi-species clocks. These are clocks that apply to both humans and mice at the same time at highly conserved stretches of the DNA. Let me start slowly. So this is the human pan-tissue clock. And I just want to give you a sense how it's been calculated. It measures methylation in 353 locations. And each location gives you a number between 0 and 1. And then you have these 353 numbers. One forms, in one way or another, a weighted average. And then this weighted average is transformed in a non-linear way so that you get an age estimate. And that age estimate is known as epigenetic age, or more precisely, DNA methylation age. And the remarkable aspect of this algorithm is that it's the identical algorithm for every tissue and cell type. It's remarkable from a biologic point of view because methylation serves the purpose of reinforcing cellular identity. If you ask, why is a liver cell different from a blood cell? The answer is epigenetics. They're epigenetic changes that control which genes are being expressed in different tissues. So in certain ways, it's a bit paradoxical, given the role of methylation, of enforcing cellular identity, that that methylation actually allows you to build an age estimator that's independent of cell type identity. Now, the pan-tissue clock is highly accurate across all these different organs. But there's a question. Obviously, in a clinical setting, it's quite easy to collect blood. And the question is that enough? And my answer is not really, it's not enough. And to show you that, I depict here epigenetic aging in different human tissues. So these samples come from a human post-mortem study. And the plot shows you what is known as age acceleration. This is really the discrepancy between your methylation age and your chronologic age. So this discrepancy in different tissues, like the upper panel shows you blood samples and bone marrow, spleen, lung, all the way to liver. And each dot is really a human person. And what you see is there's a strong correlation between epigenetic age in blood and that of bone marrow, and also the spleen. So yes, if you care about using blood as a surrogate for spleen, you will probably do very well. However, if you have good eyes, you notice that the relationship to epigenetic aging in liver samples is surprisingly low. And so overall, many organs exhibit strong age acceleration, for example, due to obesity, but blood is not a good surrogate for that. And here I show you a study that was quite interesting to me. I show how epigenetic age acceleration in different organs relates to various conditions. And the condition that was most significantly associated with age acceleration in different organs was hypertension. Obviously, hypertension is a major risk factor for many conditions, but in the past, I had a very hard time seeing that hypertension relates to epigenetic aging in blood tissue. But here you see, when I look at human fat tissue, kidney, liver, muscle, suddenly I do see a strong association. But what you should then take away is that many age-related conditions actually have a tissue-specific effect. So, for example, diabetes has a pretty strong effect on liver aging, but much lower effect on epigenetic aging in blood tissue. I studied this preservation of aging in different tissues in several animal models. Because I'm frustrated by it, I, of course, want to develop a blood-based biomarker that measures epigenetic aging in many different organs. But here I show you a study of baboons, and again, the relationships are rather weak, so same insight. Talking about primates, let's talk a bit about evolution. 66 million years ago, an asteroid hits planet Earth, wipes out the dinosaurs. And shortly thereafter, I think 58 million years ago, primates develop. And there are certain species in Madagascar that are separated by over 55 million years from our species. So vast differences in time lead to strong differences in sequence conservation. And the question is, well, can we develop an epigenetic clock that applies to all of these primates? And by the way, why would we want to have that? Well, in case we need primate models for very risky interventions, we would like to have a clock that applies to all primates at the same time. And well, I'm very happy to report that we have built such a pan-primate clock. Each panel is a different primate species. The middle panel E shows you humans. And if you have good eyes, you will see the correlation is 0.98. So our pan-primate clock is really as accurate as my old pan tissue clock, but it does apply to any primate that aging researchers care about. Aging researchers are very interested in marmosets because they live short lives, or rhesus macaque. and or vervet monkey. So yeah, these clocks work in all of them. But that, of course, brings me to an even more ambitious goal, which is, can we build epigenetic clocks that apply to all mammalian species? And for the quantitative people among you, I want to mention, what do I mean by that? So a pan-mammalian clock is a clock that is based on a single regression model. You have an aging measure in all of these species, and you then regress that age on cytosine methylation. And to develop it, you need to solve two things. First, you need to think very carefully which cytosines you look at in the DNA. And that answer is easy. Well, use cytosines in stretches of DNA that are highly conserved. You know? The next question you need to think of is, how do you deal with the fact that these species have vastly different lifespans? Bowhead whale lives for up to 211 years, a mouse less than 5 years. And so to deal with that, the easiest approach is to form a ratio. You take the age of the species and you divide it by the maximum lifespan. And as this ratio is a crude way of adjusting for differences in lifespan. That ratio is what we call relative age. So for example, if you are a 61-year-old person, your relative age is 61 divided by 122. Why? 122 is our maximum lifespan, so your relative age is 0.5. And so to address that question, I assembled a large mammalian methylation consortium. Over 200 investigators have contributed species. And here I show you pan-mammalian clocks applied to different species. So the upper left corner shows you how such a pan-mammalian clock works in humans. So quite accurate. The pan-mammalian clock works also quite well in mice, dolphins, wolves, elephants, bats, and so on. So yes, it is possible to build a pan-mammalian clock, but it's less accurate than a species-specific clock. We have clocks for dogs that are unbelievably accurate, and they would have a correlation that is much stronger than what is shown here. I want to come to a different topic. What is the age of stem cells? And now, as you know, in regenerative medicine, people are very excited about induced pluripotent stem cells to regenerate tissues. But what's the age of such a stem cell? And we know the answer. So the age of a stem cell is negative, which indicates it's a fetal age, really. And we have looked at induced pluripotent stem cells in humans, but also many other species. So yes, stem cells are perfectly young. And that gives rise to an idea, which is, can we use that procedure for generating stem cells to rejuvenate human tissues? But before I go there, I want to draw your attention to this graph. Look at the blue line. The y-axis is epigenetic age in a fibroblast, and the x-axis are days in culture after one administers the so-called Yamanaka factors, the Yamanaka factors for generating induced pluripotent stem cells. And when you look at this blue curve, you see that the epigenetic age goes down drastically within the first 2 weeks. And conversely, the markers of pluripotencies go up more gradually. And so the idea then is to say, well, maybe we express these Yamanaka factors for a short time, a week, in order to rejuvenate the epigenetic age of the tissues, but we interrupt that treatment before the cells lose their identity. Why? Because loss of cellular identity is very dangerous. It may create teratomas, cancers. Yeah, so this idea then of interrupted reprogramming has been studied by several groups. There are high-profile papers that indicate that this idea is very promising. It rejuvenates various cells, fibroblasts, endothelial cells, the optical nerve, and so on. So this is what I would call a futuristic idea. I don't know how many years it will take until these ideas can be translated into treatments that address various age-related conditions, but I just wanted to give you a glimpse of the future. I want to come to biomarkers of aging because this is obviously a very important topic for the here and now. Should we measure epigenetic clocks as part of patient care? And now the community has certainly thought about that. And one criterion on epigenetic clocks, but really all biomarkers of aging, is it must predict future onset of age-related conditions. And it must do so independent of confounders such as chronological age or sex and so on. If it has no prognostic utility, why measure it? Now, there's quite a lot of literature on that question. Do epigenetic clocks predict various diseases? And I have two answers for you. The first is definitely they do. And we know that from many large epidemiological studies. There's simply no question that on average, they do predict mortality risk and so on. However, is the accuracy enough for an individual should you go to the doctor and measure your methylation age? And that's a much harder question to address. We certainly work on it to make these clocks very robust, but I would say we are not quite there yet. But I want to mention two mortality risk predictors that we use routinely. One is called DNA methylation pheno-age. Another is called DNA methylation grim-age. And these epigenetic clocks do predict survival time. And here I give you a flavor. So the top 5% fastest agers, according to grim-age, have more than double the hazard of death than the average person. So hazard ratio is above 2 if you compare the top 5% to the average. And yeah, so this is the grim-age clock. And I just want to give you a flavor of how people construct it. So we used blood samples from the Framingham Heart Study. So these are retrospective studies. The blood was collected 30 years ago. And for each person, then we had follow-up information. We know how long these people lived, what did they die from. And so it's really then a Cox regression analysis. You regress the outcome, the survival time, for example, on cytosine methylation. And the grim-age clock is attractive because of two aspects. First is it shows a fairly strong association with mortality risk, but the second is interpretability, because the underlying cytosines actually relate to smoking pack years or various plasma proteins, growth differentiation factor 15, plasminogen activator inhibitor 1, and so on. And a lot of people get confused by that point. So grim-age is 100% a cytosine methylation-based biomarker. But it just turns out that some of these cytosines relate to these proteins, and so it gives us a glimpse of what methylation may be doing. Now we and others have, of course, related various lifestyle factors to Grimm age and other clocks in large epidemiologic studies, and the results were completely boring in certain ways. So everything that is bad, smoking, obesity, diabetes, fatty liver disease, all of these conditions are associated with accelerated Grimm age in blood. Conversely, and that was surprising to me because I was a bit skeptical, but actually vegetable consumption as measured by mean carotenoid levels was quite strongly related to Grimm age. And when it comes to exercise and education, the effects are much weaker. We can detect these effects because we have access to thousands and thousands of blood samples. However, let's say you decide you will exercise a lot and you do a Grimm age test before and after this exercise intervention. Would I see an effect? I'm quite doubtful because these effects of exercise are disappointingly low on blood methylation. And remember what I said about methylation age in different organs. Well, maybe exercise has a strong effect on skeletal muscle or cardiac muscle, but it just in blood is hard to detect. Yeah, so here I just wanted to give you a sobering look at the correlations of various factors. The middle column shows you Pearson correlation coefficients. And let's look together at the first one, mean carotenoid levels, your vegetable consumption, correlation minus 0.26, meaning it's slow. To me, this is a strong negative correlation, meaning, and I can assure you I eat a lot of vegetables. Look at education. Many of you have an MD. Was that worth it? Correlation minus 0.09, so it barely touches your aging rate, so in any event, but you see smoking, huge effect to the plus 0.44. That's by construction. The Grimm age clock actually is constructed to measure smoking exposure. Here I show you some results of relating computer tomography data that measure fat content in various organs to age acceleration in blood now. And that finding was quite stunning. You see fatty liver disease or high fat content in liver has a remarkably strong correlation with blood based Grimm age, minus 0.24. So yes, avoid fatty liver disease, This study is quite remarkable because now we look at another organ, the brain. And there are really two ways to measure the age of the brain functionally. One is, of course, to various memory tests, but also computed or various measures of brain volumes, And as expected, if your brain volumes were reduced, there was then an association again with Grimm age. So to me, that's remarkable that a blood-based markers captured that. And also, as I mentioned, memory dysfunction was associated with Grimm age. But the upper left shows you something even more stunning, which is this is the so-called G factor, better known as intelligence quotient. But what is interesting about that is that this was measured when these people were children. And whereas the blood-based measure was measured when these people were in their 70s. And so the insight is that IQ in children is actually predictive of your grim age decades later when you're in your 70s, you know, and they let you think about why that is. Probably smarter people may avoid harsher work environments, or they may avoid bad lifestyle choices. So yeah. So grim age stands out with respect to predicting time to death, time to coronary heart disease. It also relates to age at menopause. Women who enter menopause early have a slightly accelerated grim age. Want to mention it. So I like grim age, but I do want to mention that you as clinicians have many other biomarkers that are quite exciting in their own right. So blood pressure, smoking, history assessments, lipid levels, as I mentioned. So do not, and I should say blood pressure would be a far better predictor of mortality risk than grim age. It's better if you put them next to each other. But the exciting thing is that epigenetic clocks enhance these standard lifespan predictors. So they add something to it. If you measure all of the above clinical biomarkers and you add grim age in a multivariate model, they will remain significant. And the other thing I mentioned, I of course use epigenetic clocks because I'm interested in this innate aging, root causes of aging. Grim age applies, for example, to teenagers. It would be predictive of ultimate lifespan, whereas some of the others would not, perhaps. I want to talk about some topics, social status, smoking, exercise. So various groups have looked at it, and as I said, we find the expected behavior, yes, exercise. Sorry, here's a study. We looked at menopausal hormone therapy, which is controversial. Many clinicians advise against it. But what we have found is when we look at buccal epithelial cells, which in essence are skin cells, we did find that women who underwent menopausal hormone therapy, their keratinocytes, their buccal cells, were younger than controls. But our findings of blood were disappointing when we did not find a beneficial effect on the epigenetic age of blood in these treated women. Conversely, surgical menopause, bilateral oophorectomy accelerated epigenetic age in blood, actually. I looked at the female mortality advantage really in all ethnic and racial groups I could look at: African ancestry, Hispanic ancestry, Asian ancestry. And yes, women age more slowly in blood and other organs. I was quite interested in the effect of castration, but my clinical trial was rejected. So I looked at sheep. And so interestingly, in the ears of castrated rams were younger than intact sheep. So yes, castration affects at least these ear samples, but castration did not have an effect on blood. So I looked at the effect of castration in horses and in dogs, even in cats, no effect on blood. Omega-3 supplementation is a bit controversial. There were large-scale studies that have found no effect. I want to briefly mention there were a couple of publications that indicate that maybe omega-3 supplementation reduces grim age. These are very weak effects. You need to study a couple of thousand people to see anything. This was our study in the Framingham Heart Study, but there were a couple of others that have shown it. I would say the jury is still out, whether that helps grim age. Here was an interesting study by another group that looked at vitamin D supplementation. There are a couple of papers that indicate that vitamin D supplementation slows epigenetic clocks. Now I come to a clinical trial that did use epigenetic clocks. I need to start out mentioning that this was a phase 1 clinical trial that was designed to establish safety, right? A phase 1 is not a trial for efficacy. This was a trial by Greg Fahey and Bobby Brook, who developed a cocktail for rejuvenating the thymus. And that cocktail was comprised of growth hormone, DHEA, and metformin, and also vitamin D. And so these 9 men were treated for a year with this cocktail. And we looked at various biomarkers, epigenetic age, and various markers of immunosenescence. And the results were stunning, with the caveat that, so with the caveat that it's 9 people. So here I show you epigenetic age as a function of months in the trial. And in the lower left, you see Grim age. And what you notice is that this cocktail rejuvenated Grim age by 2.5 years, which to me is a remarkable success. For the people among you who are in their 80s, they will say 2.5 years is not good enough for me. But in essence, it shows that this cocktail stopped aging in blood. So the good thing is Greg Fay and Bobby are now conducting now a phase 2 clinical trial, and hopefully we will have some results in a year or so. Before I move on, I want to mention that we do need these results from the phase 2 clinical trial, because the cocktail is a bit controversial. It involves growth hormones injections, and you as clinicians know that there are certain risks with that. I want to move to an anti-aging intervention that is non-controversial. It's caloric restriction in mice. And there's no doubt that that intervention slows all of our epigenetic clocks for mice, no question. And so in my lab, we study many of these gold standard interventions. We can discuss them later, but I'm interested in all interventions that are supposedly slow aging. But one question is, what is the meaning of epigenetic clocks? As I mentioned, the original sin is that we need to work quite hard to understand the biology. And I want to draw your attention to a scientific paper by Ken Raj, who looked at various hallmarks of aging. And the first hallmarks are quite famous: cellular senescence, telomere attrition, and genomic instability. Interestingly, when we looked at cells in a dish and fibroblasts and keratinocytes, we really don't see a connection between epigenetic clocks and these hallmarks of aging. However, there are other hallmarks: mitochondrial dysfunction, stem cell exhaustion, deregulated nutrient sensing, and altered cell-cell communication. They do relate to epigenetic aging. So I just give you a glimpse of one of these experiments that Ken carried out. So these are different cells in the dish, and Ken induced cellular senescence in 3 different ways: through radiation, gamma rays, or RAS oncogene induction. And they were not associated, these interventions did not accelerate the epigenetic age of the cells. However, replicative senescence, if you passage cells in a dish, then they become ultimately senescent. That phenomenon is known as the Hayflick limit. And that was associated with an increase in epigenetic age. But the reason is for that latter finding was that the cells were cultured for many months. And so just through because 6 months passed, these cells aged. It did not reflect telomere shortening in any way, so. And one way to see that telomeres don't relate to epigenetic aging is to immortalize cells. You can do that by telomerase or TERT overexpression. And that does not arrest epigenetic aging in the dish. I mentioned nutrient sensing is strongly linked to the epigenetic clocks. There are many experiments that show it. For example, rapamycin decreases epigenetic aging in keratinocytes, but incidentally, only keratinocytes, not in blood. But I mentioned caloric restriction lowers epigenetic age in mice. In humans, I found that obesity greatly accelerates the epigenetic age of human liver samples. So yes, that very much relates to epigenetic aging. Mitochondrial function does relate. So various interventions that improve mitochondrial function or conversely impair it are associated with epigenetic aging. The results are fairly strong, but occasionally our experiments fail. So occasionally we disrupt mitochondria and it does not affect the epigenetic clocks. The one finding I'm very sure about is that HIV infection accelerates age. I looked at multiple organs, I looked at brain, I looked at blood, kidney, and that's very reproducible. So yes, HIV infection is associated with epigenetic aging. Lately, there were a couple of papers on COVID infection. I remember 2 papers that showed that COVID is associated with increased grim age. But I also saw a paper that did not find an effect, so I think we need large sample sizes for these studies. Briefly want to mention centenarians. I looked at Italian centenarians. So yes, if you have parents who are centenarians, chances are that you inherit their longevity genes. And yes, these longevity genes are associated with lower epigenetic age. I'm quite confident in that finding because I looked at Italian centenarians, but I also looked at data from the so-called UK Biobank, several 100,000 people, and we found similar associations, meaning if your parents lived a long life, your grim age is lower than that of people who are controls. Some people are interested in stress. My impression is that long-term stress, severe stress is associated with age acceleration. What do I mean by that? Sexual abuse in children leads to severe stress. That was associated. Here I show you a very nice study of post-traumatic stress disorder in veterans that also found an association with grim age, but not the other clocks. This is one of these findings where you really need to think, what clock do you use? Sleeping, the expected behavior, and this is a study of the number of insomnia episodes per night. And yes, certain clocks, not all, but certain clocks pick that up. If you have disrupted sleep, for example, these blood-based clocks show age acceleration. This is an interesting study I want to show because let's step back. I would love it that epigenetic clocks find clinical utility. That's my dream, that you as clinicians find them useful. And so I'm looking for conditions that may provide an avenue. And so here is a nice study of atrial fibrillation, where the investigators looked at epigenetic age acceleration in grim age and found that, yes, higher hazard rates could be observed in people who ended up developing incident atrial fibrillation. And that was a large study, multi-ethnic, over 5,600 people. Here's another condition that in my opinion is probably the most promising for epigenetic clocks. And that is known as clonal hematopoiesis of indeterminate potential, CHIP, or also known as age-related hematopoiesis. So what is that? As we age, we incur certain mutations in DNA methyltransferase or TET enzymes, and these somatic mutations are associated with much higher risk for various blood cancers. And interestingly, there were two papers that showed that these mutations, this CHIP is associated with epigenetic age acceleration. I show you here just one graph that shows various epigenetic clocks, and all of them were associated with increased epigenetic age acceleration in people who had these CHIP mutations. That's finding one. But the finding two is far more exciting from a clinical perspective, which is that the epigenetic clocks allowed you to stratify the patients by risk categories. So the left panel shows you all-cause mortality risk. The purple graph shows you the probability of death in people who had these CHIP mutations and exhibited epigenetic age acceleration. So they are at the highest risk. And conversely, when you look at the turquoise graph, these are people who do have the CHIP mutations, but who do not have any epigenetic age acceleration. And you notice they don't have increased risk, you know. So this is a stunning finding because it suggests that, let's say you have a patient who has these CHIP mutations, you would then send them to an epigenetic clock testing facility, and they would say, yes, you are in big trouble because you have epigenetic age acceleration. Or conversely, you could perhaps tell your patient, don't worry about it, you don't have age acceleration. I want to switch gears and talk about progerias. There are many different progeria types or segmental progerias. If you ask me, according to epigenetic clocks, Down syndrome is the #1 progeria, is associated with increased age in brain, in blood, several tissues. Perhaps also Werner syndrome. And finally, cocaine syndrome. So my epigenetic clocks imply that these three progeria are associated with increased epigenetic aging. I want to briefly mention, we studied Down syndrome in a mouse model, and even in that mouse model, did we observe epigenetic age acceleration. I think I've shown you that our epigenetic clocks are being used in a couple of clinical trials, but I'm not yet satisfied with the state of the art. We want to develop so-called causal clocks. And what do I mean by it? Well, let's say you do have an anti-aging intervention that reverses the age of a causal clock. You then can actually tell the patient, yes, you are likely to have a clinical benefit, you know. And in order to develop such causal clocks, we really need to increase our mechanistic understanding and simply do more work, you know. Now, I should tell you, in mice, we have solved the problem. We do have very, very convincing causal clocks in mice. They track all of these aging interventions that I mentioned, but there are a couple of more. So gold standard interventions in mice are so-called growth hormone receptor knockout. You get a dwarf mouse, and sure enough, that is associated with slower epigenetic aging. There's a more controversial perhaps finding, which is this idea of young blood parabiosis. You inject young blood, but that also is associated with younger epigenetic aging. Conversely, several progeria models show the expected behavior. With rapamycin, I'm still on the fence. We have some data that indicate that rapamycin is beneficial. I mentioned human keratinocytes, but I analyzed human blood samples and I didn't see a beneficial effect of rapamycin. Oh, yeah. So I conclude just some take-home messages. We do have several epigenetic clocks. They do serve different purposes. Lifestyle affects only some of these clocks, not all. Clocks also differ in terms of predictive accuracy. For example, my pan-tissue clock is a bad mortality risk predictor. It does predict mortality risk, but it's a bad one. Conversely, grim age is a much better risk predictor. I also mentioned several times, none of these biomarkers will ever replace what you currently measure in the clinic. They complement it. Yeah. And if you design your own study, you really want to think very carefully about the source of DNA. Do you want to draw blood? Some of the interventions have a much stronger effect on buccal swab samples. I mentioned menopausal hormone therapy. I would also venture to say that rapamycin, various hormone treatments probably have a stronger effect on keratinocytes because keratinocytes have more hormone receptors, so they pick up these effects. But you can measure epigenetic age in urine samples. Urine has enough DNA, fat tissue, muscle, skin, and so think about it. Yeah, so this is my acknowledgement slide. I have started a non-profit research organization called the Clock Foundation that is meant to facilitate rigorous testing of epigenetic clocks. But I'll stop with that. Thank you so much. Yeah.
I want to thank you, Dr. Horvath, for taking so much time to spend with us today. We really appreciate it. The presentation was just fascinating, and I hope that we can follow up with you and circle back and get your advice and your guidance on, some of the data that we should be collecting here, and maybe work with you and collaborate with you on that.